In this chapter, we're going to be talking about cell reproduction, and it's going to lead us into our genetics unit, which is going to come up next. So, as an overview, we're going to start with this little thought. Cancer. Cancer, obviously very dangerous, can be deadly. What is cancer? Well, cancer is really cells that are going through uncontrolled cell division. They're dividing so much that they're becoming things like tumors. They're disrupting the function of cells around them. And so when we think about cancer, we have to think about cell division. You know, why do cells divide? What causes cells to divide in an uncontrolled way? And how do our cells normally control their division? And so the overview of this chapter, we're going to talk about how cells divide, different ways that they do this, the cell cycle, meaning the stages of life of a cell. We're going to talk about mitosis, which is a type of cell division. Then we're going to talk about meiosis, a different type of cell division. And then we're going to talk about what happens when things go wrong. We'll talk about cancer, and we'll talk about abnormal numbers of chromosomes and things like that. All right, so let's start by talking about cell reproduction. Why do cells reproduce in the first place? Well, there's really three reasons. Number one, they allow the organisms to grow, right? You're made of way more cells than when you were born because you've made more cells to get bigger. To replace cells that become damaged when you get a cut or when cells just get worn out. Skin cells, for example, only live about 35 days. They work their way to the surface and then they sort of float off and become dust. And we have to replace those cells. And to make more organisms. I mean, simply to reproduce and pass on our genes. And why can't we just be made of a few really big cells? And we talked about this actually in an earlier chapter, but we'll revisit it one more time. So the volume of a cell increases about 3 times faster than the surface area of the cell. So what does that mean? Well, the surface area is the distance around a cell. So if you think about the surface area as all of this exposed surface around the cell, and the volume is how much stuff is contained inside. Well, the bigger a cell gets, there's actually less surface compared to this volume inside. It gets bigger much faster than the surface. And what that means is that as the cell gets bigger and bigger, the distance that nutrients would have to travel to get to the middle and the distance that waste would have to travel to get out increases. And it can become such a critical distance that the cell can't maintain itself. It can't stay alive. And so this is why our cells are limited in size. They basically can get too big to where the nucleus can't control it and to where nutrients can't get to the center, wastes can't get out fast enough. So our cells have to divide. So cell division is the way our cells come about. How do new cells come about? They come from other cells. There are 2 different ways that cells can reproduce. They can reproduce asexually, in which case we're talking about offspring that are exact copies of the parent, only require one parent, and this is how most of your cells are dividing. Your skin cells, your bone cells, your blood cells. They are basically just making clones of one another. Bacterial cells also reproduce asexually. Some small organisms reproduce asexually. On the other hand, we can also reproduce sexually. Sexually means that there are 2 different parents combining to make an offspring. And so the offspring are actually going to have variety. They're not going to be a clone of either parent. They're going to be a combination of the 2. And we're going to talk about both of these in this chapter. Now, cell division in an amoeba. Well, we talked about 3 reasons cells divide: to replace cells that die, to repair, and for growth. And then to make new organisms. Well, an amoeba is a single-celled organism. So when its cell divides, you basically get 2 amoeba. So this would be for the purposes of reproduction. To make more organisms, you would go from 1 amoeba to 2 amoeba, and 4 and 8, and whatever. But in your body, our reasoning is different. This is more of replacing the cells that die and also allowing for growth. right? The cells in our body on a normal basis are not reproducing to make new people, but to replace cells that die and allow us to grow. This is just showing some examples of asexual reproduction. So in yeast, this is just a simple division. In a sea star, fragmentation, if you cut off one of its legs, that leg will actually grow into a new sea star. And here, cutting, you can basically cut the African violet, and then the piece will grow into a new flower. And of course, sexual reproduction gives us variety, whereas all of these are going to be, this sea star is going to be a clone of the one it came from. These two are going to be clones of each other. This leaf is going to have the exact same DNA as the original plant. On the flip side, sexual reproduction gives us variety. So all of these people, nobody's going to have identical DNA to anyone else. Everyone's going to be combinations. All right, prokaryotes, the way that they reproduce is something called binary fission. Binary fission is literally splitting in half. Remember what a prokaryote is, by the way? That's a bacteria. And what's really going to happen, they usually have, if you look at a bacteria, they have one circular chromosome. This is their DNA. It's a circular one chromosome. It's a double, imagine this being the double helix. And what's going to happen is this chromosome will make a copy of itself, and then this entire bacterium will split, and you'll get two separate bacteria, and they will be clones of one another. It is asexual reproduction because one parent and the two offspring that are made from that one parent are exact clones. Here's a picture of it. So this is the circular chromosome. It looks like a big blob, but if you were to stretch this out, it would literally be one loop of DNA. So this is a loop of DNA. And what it's going to do, it makes a copy of itself. The whole thing splits in half. And this can be as quick as about 20 minutes. So bacteria reproduce very, very quickly because it's a very simple process. All right, let's talk about eukaryotic cells. Now, eukaryotic cells are a bit more complicated. We're going to skip over here. So in us, we're not talking about one chromosome. We're talking about multiple chromosomes. In fact, if we're talking about humans, each of our cells has 46 chromosomes. Also, our chromosomes are not circular loops of DNA. Our chromosomes are linear. They're a straight line of DNA. It literally does have a top and a bottom. Now, when the cell is not dividing, this is what they're talking about. If you're looking through a microscope, if the cell is not dividing, you would not actually see chromosomes. You would just see inside the nucleus something that looks kind of like this, and that's called chromatin. And what that is that's the DNA all sort of unwound into its longest possible scenario. When the DNA coils up, when the cell is going to divide, what will happen is, if you imagine this is the chromatin, and this coils up super, super, super tightly, and that's what becomes a chromosome. Usually that also actually makes a copy of itself, so you end up with two sides. And so we usually draw the chromosomes like this, as these X's. But in reality, what you would see if the cell wasn't dividing, you would not see this. This would be unwound into just a bunch of like string. It would look like just a big loopy wad of string. Now, here's the thing. Before the cell divides, okay, a cell has to make a copy of its DNA. So if you were to take a cell, if you were to look at, say, your cell when a cell is first born and it has 46 chromosomes, they would look like this if you were to coil them up. They would look like sticks, and you would have 46 of these. 46 of these sticks. Technically, they would be in matching pairs. There would be two of each one. And then what's going to happen is before the cell gets ready to divide, since it's going to give a copy of its DNA to each of the offspring, each one of those sticks would make a copy of itself. So then these chromosomes are now going to look like X's. So now, instead of 46 sticks, you would have 46 of these X's. So there would still be 46 chromosomes. This is drawn very poorly. So imagine there would still be 46 of these, but instead of 46 sticks, they would be 46 of these X-shaped things. Now these two sides are called chromatids. So this is one chromosome before it makes that copy of itself. and then it makes a copy of its information, and now these two sides, this is still one chromosome, but these are called sister chromatids, and it turns out that these sister chromatids have the exact identical information. That way, when the cell divides, this can split in half, and each new cell would get an exact copy of the info. All right, so when does this happen? When does it consist of two chromatids? Well, we haven't talked about it yet, but we're going to talk about something called the cell cycle, which I'm going to draw. So this is when the cell divides. I'll just put division here for now. We're going to get into more detail later. And all of this is called interphase. This is when the cell is not dividing. And it turns out that is subdivided into three phases, G1, S, and G2. And so to answer this checkpoint question, when we're about to talk about this, the cell would have chromatids here. S stands for synthesis, and this is when the chromatids would form. So here in G1, the chromosomes would look like sticks. In S, they would now look like this, like an X. And that would be the same in G2, and then the cell would divide, and they'd go back to being sticks again in the offspring. So, this is a cell getting ready to divide. See this? These are the chromosomes actually forming from the chromatin. So they're coiling up into these tight little rods. And what this is showing, so this is a chromosome before it goes through that stage we're going to talk about called S. This is now the two sides, the chromatids. They're held together. And then when the cell divides, this would split back in two again. So, the cell cycle is a sequence of events, and it starts from when the cell is first formed, and then goes through the cell dividing to the next generation, so to speak. If a chemical prevents DNA synthesis, this would trap the cell. Again, they're asking this question, and we haven't talked about the cell cycle, but the answer is going to be it would be trapped in the first stage of the cell cycle, which is called G1, and it would never be able to go past that. So this is what the cell cycle looks like. So you have interphase is all of this. This is interphase. You can see they have it labeled here. It's subdivided into G1. It stands for first gap because they used to think nothing happened, so they called it gap phase 1. S, which stands for synthesis. Again, this is where the chromosomes make copies of themselves. Now they look like X's. They're duplicated chromosomes. Same number. You don't double the number of chromosomes. In our cells, you would have 46 chromosomes here. You would still have 46 here, but now they're 46 X's instead of 46 little sticks. G2, gap 2, stands for gap 2. The cell would prepare to divide. And then mitosis. This is where the cell would actually divide, make a copy, and then you would start the whole thing over. This is actually a better picture of it. So here's interphase. G1, S, G2, and then this is mitosis, the steps of mitosis, where the cell divides. And really quick, so mitosis is going to be the process where the nucleus divides in two. It's going to form what's called a spindle or spindle fibers. And the sister chromatids, what's going to happen is they're going to line up down the middle and attach to these spindle fibers, and they're going to get pulled apart. And that's why the chromosomes are going to go from being X's in the new cell. This will now split into two cells, and each cell would have identical information. So, stages of the cell cycle. So, interphase is the entire time when the cell's not dividing, most of the time. G1, primary growth. It stands for gap, but really I think of G in growth. The organelles can divide, the cell gets bigger. S, this is where the chromosomes go from being a stick to being an X. Even though you can't see them, right? It still looks like what's called chromatin at this time. You wouldn't see them. G2 stands for gap 2, but technically the cell's preparing to divide. Mitosis is technically division of the nucleus, but at the very end, cytokinesis happens, and that's where the remainder of the cell splits. So this is the cell cycle, and that's actually where we're going to stop for today. We'll start with mitosis next time.